heartwarming words or welcoming thing that brings lot of hopefulness. Now, may I request Dr. Mary Baptista Janet, Associate Professor of Zoology and Dean, School of Biological Sciences, to accord the warm words of welcome. Over to you, ma'am.
Yes. Very good afternoon to everyone connected here. I'm delighted to be connected with you all on the third day of this faculty training program to welcome and introduce Dr. Baby Lakshmi Muthuswamy, Research Scientist, Institute of Bioinformatics, Bangalore, who is going to enlighten and update us on computational approaches for microbial diagnostics. Dr. Baby Lakshmi has attained her bachelor's and master's degrees in physics from MS University and got her advanced diploma in bioinformatics from Madurai Kamaraj University. She has done her doctorate in bioinformatics in Pondicherry University. Ma'am has got over 20 years of research experience in various biological data analysis, designing and development of biological databases. With her expertise in bioinformatics, she has started her career as a scientist in DSQ Biotech Limited. She has worked as research scientist in computational biology, genomics, and proteomics at the Institute of Bioinformatics, and as an adjunct faculty in Manipur Academy of Higher Education. And at present, she is serving as a research scientist in computational biology, inherited genetic disorders, genetics, genomics, and proteomics at the Institute of Bioinformatics. Dr. Baby Lakshmi has proficiency in analyzing exome sequencing data, identifying inherited genetic disorders, Python programming, shell scripting, R programming, analyzing proteomics data, and protein expression patterns. She has trained herself in developing database using MySQL and web development using Zoho. With her vast knowledge in bioinformatics, she has authored and co-authored around 36 research publications, including articles in the top-tier journals such as Nature, Nature Genetics, Genome Research, Proteomics, Molecular and Cellular Proteomics, etc. Ma'am has played a vital role in the development and maintenance of internationally renowned biological databases. She has never missed the chance to enthrall the audience through her informative lectures, through workshops conducted at the Institute of Bioinformatics on NGS data analysis. With her research experience, she has completed a project worth 40,45,000 funded by DBT and handling 5 more projects funded by ICMR and DBT. She has been honored with the fellowship awards by CSIR and Department of Biotechnology, Government of India. She has won second prize for developing a scientific application titled Research Pathways in Elsevier, Code for Science Competition, India. Dr. Baby Lakshmi has presented a poster entitled A Novel PAK3 Variant in an Indian Family with Non-Syndromic X-linked Intellectual Disability at the International Conference of Indian Genetics Congress 2015, which won the Best PhD Research Award. So I feel that we are all singularly fortunate for getting this chance to listen to such an eminent scientist today. On behalf of Marian Star Center, PG and Research Department of Zoology, St. Mary's College, and Department of Zoology, Rani Government College for Women, Tirunelveli, I accord a warm and cordial welcome to you, ma'am. I'm happy to welcome our Vice Principal, Dr. Sister Shibana, the overall coordinator of STAR program, Dr. Sister R.K. Genesius, our Controller of Examination, Dr. N. R.K. Mary, the head of our department, Dr. Hermin Pasanya, and the heads of various departments to this program. I extend a genial welcome to all the esteemed professors who are participating in this training program. Once again, a hearty welcome to you all connected here. Thank you.
Thank you, ma'am, for entangling us with your heartwarming words of welcome. It gives me immense pleasure to invite Dr. Babilakshmi Muthuswamy, Research Scientist, Institute of Bioinformatics, to deliver the lecture on computation approaches for microbial diagnostics. Over to you, ma'am.
Thank you very much. Thank you for the kind introduction, and I'm happy to be part here. And it would be really great if I can be in person and present in front of you all. But because of the pandemic, I think we have to admit all these things, and then we can go ahead using these like advancements in computation for these purposes. So I would like to start. I would like to share the screen. Hope my screen is visible.
Yes, ma'am, it's visible, ma'am.
So I'm doing slideshow. Is it visible now?
Yeah, it's visible, ma'am.
Okay, then I'll start the presentation. Once again, thanks for the opportunity. And I'm Dr. Baby Lakshmi from Institute of Bioinformatics. So I am a research scientist. I'm going to talk about the computational approaches for the microbial diagnostics. It will be like a brief introduction. The microbial diagnostics is basically used in the major area of infectious diseases to identify the cause for the diseases. So it is caused by the presence of pathogenic microorganisms in humans that are able to cause the disease. So it get transmitted, the infectious diseases that get transmitted from one species to another. And the pathogenic microorganisms can be bacteria, viruses, fungi, protozoa, and multicellular parasites. So the clinical diagnosis is nothing but to identify these pathogens in the species. So to start with, the patient arrives at the clinic and the samples will be collected. The sample sources can be urine, sputum, CSF, etc. Morphology, many potential pathogenic species will be identified. These things are happening in the routine clinical diagnostic laboratories. So, in case of some pathogens which are very meager in quantity, which requires culturing. So, for example, Mycobacterium tuberculosis requires culturing to identify in the microscopes. So, so we have to culture them and then detect the pathogen. And other methods are like some immunologic assays like latex agglutination and ELISA are some of the methods to detect the antigens, and PCR based methods to like Sanger sequencing and PCR based methods to from the DNA and RNA of the samples, we detect the specific pathogen, whether it is present or not. And also antibody demonstration. These are some of the methods routinely used in the clinical diagnostics laboratories, and it is laborious and time consuming. Also, a question is whether are they accurate? Most of them are accurate, yes, but some of them leads to confusion, and that affect the treating the patients. So these microbiologists do the laboratory techniques and then identify the pathogen and report to the clinician saying that these are all the pathogens found, and based on which the clinician treat the patients. So the current method is to avoid these laborious techniques. The next generation sequencing methods are becoming very popular recently, and The traditional methods of identifying the organisms, such as these pathogens, are often very slow, and specialized techniques are needed. And some organisms, the organism to organism techniques differ. So the NGS has revolutionized the clinical diagnostics, and it replaces the current time-consuming and labor-intensive diagnostic methods. It is so these next generation sequencing methods are very advantageous for the microbial research as well as the microbial diagnostics. So I'll give a brief introduction about what is next generation. I'm not sure the audience whether they have know well about the next generation sequencing methods or not, but I'll give a very brief introduction, and then we'll move on to the real work we have carried out at our laboratory. Sequencing, this is a sequencing chronology. First, DNA was discovered in 1869, and then Sanger sequencing, which was the first sequencing machine, was invented. The method, Sanger sequencing method, was the first method for the sequencing any DNA, RNA, or was identified in 1977, and then first automated sequencing machine was invented, and then whole human genome project was completed with that Sanger sequencing method itself. And in 2005, first next generation sequencing machine was invented. And in 2013, there were 10,000 complete genomes deposited in NCBI because of the advent of these next generation sequencing technology. In 2017, there were 73,996 complete genomes in NCBI, and now it is increasing enormously. It is increasing, and the number is keep increasing. And the current number I do not have right now. So this is the representation, graphical representation of the increase. Not only specific particular eukaryotic species is being sequenced, but also based on the need, different bacteria, fungi, and mitochondrial genomes, plasmids, protozoa, and many other genomes, a diverse population of genomes are being sequenced, and the complete genome is available, and these are stored in the database called NCBI. So we can download these sequences and use it for our purposes. So next generation sequencing, the basic idea how it works, I am giving a brief introduction on this. First, the DNA will be, genomic DNA will be isolated, and the DNA will be fragmented into small pieces, small fragments. For example, 500 base pair long fragments will be like cut the DNA into 500 base pair long, approximately 500 base pair long fragments, and then we attach adapter. These adapter sequences are small structure sequences that actually binds to the a flow cell on which sequencing takes place. So the flow cell will have a complementary sequence embedded in this, and then after we provide these fragments on the flow cell, then hybridization takes place, and then these fragments are amplified, and clusters are formed. Like it is mentioned, bridge amplification is taking place, and some clusters are formed. The cluster 1, 2, 3, 4 are shown in the figure, and these clusters are further sequenced. They are sequenced in both the ends. Each fragment is sequenced in both the ends. These fragments are cut such a way that overlapping fragments, so that we will not miss any information. So we sequence it on both the ends. That means that is called as parent sequencing, and the sequenced product what we get is called sequencing reads. So basically, while sequencing, we get the digital image, colored images for each nucleotide, and that will be decoded, and then we get the sequences. Further, it is aligned to the reference sequence, and then we do further analysis. So some of the sequencing technologies, different technologies were used, and different companies have invented these technologies, and different machines were produced. So Illumina sequencing is very popular, and it's being widely used by several people for various purposes. And 454 pyro sequencing was quite old because of these are error prone, so it's not used much. The Illumina sequencing can produce up to 150 base pair at a time. When we sequence the fragment, it can sequence up to 150 base pair at a time. So we do sequencing of both the ends. So 300 base pair of the fragment will be sequenced. Then pyro sequencing can sequence up to 200 to 400 base pair. Solid sequencing, it's a small fragment only it can sequence, and iron torrent is another method. So these machines generate high throughput data. It is possible to do sample multiplexing. That means at a time while sequencing, you can produce, you can provide multiple samples, and then in 1 or 2 hours, it generates the results. While multiplexing, they add a barcode so that you can separate out which samples, which reads belongs to which samples. And then it's a paired sequencing. That means on both the ends both the ends of the DNA fragment is sequenced. It's becoming cheaper day by day, and it is made affordable to the normal research labs. So next is third generation sequencing technology. It is recently developed technologies and still under continuous development. This includes PacBio sequencing and nanopro sequencing. They also use two different chemistry to sequence these molecules. These two methods follow the same principle of sequencing one molecule at a time, and they have the capacity to sequence up to 15 MB fragment, 15 MB long sequences at a time. So that is the major advantage of these sequences. And the previous ones, short read sequencing methods, they have capacity up to 150 base pair sequencing. So when we align it back to the human genome, kind of large genomes, some regions where repetitive sequences are there, those regions are like wrongly placed. So 15 MB is quite unique pieces to align it back to the reference genome. So this is more advantageous. One of the disadvantages, these sequences produce more erroneous bases. So we have to be very careful while analyzing the data. So these are mainly used for the detection of the pathogen, and there are many tools that understand the errors in these tools, and then in these machines, the reads generated by these machines. So the assembly and analysis becoming easier. So these technology generate millions to billions of short read sequences. It can do whole genome sequence, it can do targeted region sequence, and it can also do RNA, transcribed RNA sequencing. So these has to be analyzed to get the meaningful insights of the data. So it can be aligned to the reference genome. That means we generate from a sample, we generate some reads, and we have to make meaningful insights, and we can either if the organism of our interest is already known and the sequences are already available in NCBI, then we can download from the NCBI, and then we can align it with the reference genome, and then we can find what are the differences. And in case if the species is not known, its sequences are not available previously in NCBI, then we have to assemble these fragments into single fragment, and then make out which species, which similar species it can be, and make the analysis. So this is just to show, this is a quality control. It's like it's an image for the quality control. So we use a program called FastQC. Now it generates several measures of the quality of the sequence reads. So this is one of it, to understand the per base sequence quality. So x-axis contains the positions of the read. The position like 1 to, it can go to 100, or 150, it can go up to 150. Each read fragment is 150 base pair long. So here it shows up to 100 base pair. At first position, what is the quality? Quality is Fred scale quality. If Fred score is 10, then it is there are errors in 1 in 10 bases. So it is 90% accurate. And if it is 20, it is 1 in 100 bases or yeah, erroneous. So it is 99% accuracy. So we generally consider 20 and above are good quality reads. So this is showing the y-axis contain the Fred scores. So it has 20 and above. So this data is good quality data. So mostly Illumina sequencing, it saturates towards the end and it goes down. So in case if needed, then we have to trim those 3' end bases and then proceed with the analysis. So the quality is very important. If the some of the reads are low quality, we have to discard them. So after doing the quality control, this is another depiction of how the alignment will look like. So this is aligned to the human reference genome. This is the top row, the top line represents the chromosome. And after, as I said, it's a overlapping segments, and each fragment is amplified, and it is sequenced several times. And because of that, we are seeing several reads mapping at particular position. You can see that at one particular position, we see several reads. So I'm not sure whether you are able to see the mouse pointing, but these are all, each line represents one read, and it starts and ends and overlapping reads so that it covers the entire region of the genome. This will just show. So after doing quality control, we align it to the reference genome, and then we identify what are all the variations compared with the reference genome. So what are all the variations we look for? It can be SNVs, single nucleotide variations. It can be small insertions as compared to the reference genome, and it can be a small deletion, or it can be substitution. So a stretch of nucleotides are substituted with another stretch. And even translocation, inversion, copy number variations are some of the advanced some of the additional variants found in several species. The sequencing data. What are the applications of the NGS and bioinformatics in microbial diagnostics? It's like I would like to point out only very few, which we routinely do in our laboratories. It's majorly used in microbiology to study the infectious diseases. To detect the pathogen and identify the drug resistance causing mutations and the associated genes, and the host pathogen interactions. And also it is used in vector biology. For example, Lyme disease, it is caused by tick bite. If the tick has a Borrelia burgdorferi, then it causes the Lyme disease in the human, where if the tick has Borrelia burgdorferi, then it causes Lyme disease. And malaria is caused by Plasmodium falciparum. The mutations and the presence and absence of these pathogens can be studied. And virology also, it detects dengue virus, Ebola virus, mutations in these sequences, Zika virus and coronavirus, and everything can be done with the NGS sequencing. Basically, to isolate the DNA or RNA based on the virus type, and then we do the sequencing, and then compare it with the reference genome, and then see what are the differences, what are the variations that occur, whether it is associated or whether it is causing the disease or not. So at our institute, I would like to talk about two or three, two if time permits, and then I will. If time permits, I'll go into the third one. Three projects that we have done using the microbial diagnostics using next generation sequencing methods. So this one is whole genome sequencing we have done on 200 pulmonary TB clinical isolates to identify the drug resistance causing mutations. So we have collected samples, and the Mycobacterium tuberculosis clinic was isolated, and then we have done the sequencing. The first we did the drug resistance profiling using drug resistance sensitivity test, and then we found that it is sensitive to first line drugs. There are 50 of them, and monoresistance with rifampicin, streptomycin, isoniazid. They were 12, 6, and 31, and multi-drug resistance were 91, and pre-XDR were 10. Totally 200. We have isolated Mycobacterium tuberculosis, cultured, and then we have done the sequencing using the Illumina platform, and then further data analysis was carried out. First, we have to do the quality check. As I said, FastQC is the tool used for quality check, and as I said in the beginning, there are some adapter sequences attached to the fragments, so we have to remove them. And if the quality of each base is below 20, then we have to trim it. And then mapping against H37RV, which is the reference genome of Mycobacterium tuberculosis, and then using the BWA algorithm. And then there's a technique called local realignment, so that we can identify the indels and duplications accurately using the tools called GATK and Picard. GATK and Pindle are the two tools that use to identify the single nucleotide variants. Single nucleotide variants and insertions and deletions. So if variants with greater than 5 reads supporting with a base quality score of greater than 20 will be considered, the rest of them will be discarded. So annotation of the variants we carried out using our in-house scripts. So these are all the results. So basically the drug resistance associated genes are previously already known. For example, isoniazid associated drug resistance causing genes are CATG and INHA-BC, and rifampicin drug resistance causing genes are RPOC ABC. And some mutations are already known. They are already known that they are causing the drug resistance. So basically, we have to, in these 200 isolates, we have aligned to the reference genome and then identified the variants and found that which are all the mutations found in these drug resistance causing genes. So for rifampicin, isoniazid, and ethambutol, these are all the variants we identified, and we also found that, for example, CATG or 463 to L that is arginine converted into leucine at position 463 is found in more than 140 clinical isolates. That means 140 of them are isoniazid resistant. So like that, we have these many mutations across different samples. So this is the results. This is a routinely we look for these mutations, and we also found 343 novel single nucleotide variants and 38 drug resistance causing genes, which are not currently used in the diagnostic kits, because our diagnostic kits generally look for very limited number of mutations, and that are really known that drug resistance causing mutations. So the whole genome sequencing of these species have the potential to identify all possible mutations that may that can cause the resistance pattern. So in addition, we have also found that 20% of these clinical isolates are with co-infected with the non-tuberculous mycobacterium species. So that means we have also identified non-tuberculous mycobacterium species in these samples. So around 40 samples we have identified this. This figure shows that different organisms, the amount of percentage of different organism or different species found in these isolates. In this Mycobacterium tuberculosis is in red color, so that shows the load, how much it is present, and we see that, and also the other colors represents the other non-tuberculous mycobacterium. So we also see that there are some isolates, there is no red color, that means no Mycobacterium tuberculosis, it is only other non-tuberculous mycobacterium. Example, this green color, the three green color ones, that shows Mycobacterium africanum. This is accurately detected by the next generation sequencing methods, which cannot be possible with the routine clinical diagnostic methods. So this is another advantage of next generation sequencing method. So to conclude that, we have done the accurate profiling of drug resistance causing mutations and accurate detection of the pathogen, and we have also identified the mixed infection, and we have identified the presence of only the non-tuberculous mycobacterium in certain samples. So I'll move on to another project we have done on Cryptococcus to directly identify the Cryptococcus from CSF, cerebrospinal fluid, from meningitis patients. So for the interaction, it's encapsulated yeast and obligate aero that can live both in plants and animals. It causes cryptococcosis in immunocompromised host organism, and it affects the lungs, and meningitis and encephalitis is also seen. So if untreated, it causes the confusion, reduced level of consciousness, seizures, and it can lead to coma also. So we have taken three samples, three Cryptococcus positive CSF samples. We have collected it from a Department of Neuromicrobiology at NIMHANS. So we have done the routine laboratory techniques. We collected the samples, CSF samples, and then routine clinical microbiology workup was carried out, and it is cultured on SD agar plates, and we found the creamy white colonies, and India ink staining was performed, and we detected the Cryptococcus neoformans in these three samples. Further, we further centrifuged the CSF samples and then lysed the cell walls, cryptococcal cell walls, and isolated the DNA. Then a library, we have to prepare a library so that it can be sequenced. We have used nanopore sequencing. It is a single molecule sequencing method. So one by one molecule it will allow and sequence. We have done that. Some of the laboratory experiments were carried out. These are the methods. India in test was positive in all the 3 samples, and bacterial culture, there was no growth in bacterial culture. Fungal culture was positive in 2 of them, and different additional laboratory experiments were done. So the data analysis was carried out, nanopore sequencing data analysis. The raw data, the quality passed reads only will be considered. And we have converted this, it generates a file called fast file, and then it will be converted into FastQ, which is a routine, which is with that only we used to do the routine analysis. And ionize R is the R package that is used to demultiplex. So we have used 3 samples, we mix it and then do the sequencing in a single run. So this is cost effective. So you are running the machine only once for the 3 samples at a time. So we have a barcode. We have to using that barcode that is called separate the reads. That's called demultiplexing. Then we have used we have done taxonomic classification using last one tool, and also the Patrick. Another tool. These do the taxonomic classification. What does that mean is whatever reads it was generated, it will compare it with the already known sequences, including all the known complete genomes. It will compare, and then it will report how many reads find to how many organisms, how many reads map to how many organisms. It will give the listing of reads and number of reads mapping, the organism and number of reads mapping to it. And also we have done, because we have found the Cryptococcus neoformans was expected in these samples. So we did aligned it against the Cryptococcus neoformans reference genome itself. And then we found how many reads are matching, and we have also found how many reads mapping at particular position, that is called depth of coverage, and also how much of the genome is covered with the reads, that also calculated using samples. So further, we also did a blast analysis that is we take the sequences and run it against the NR database, and then we identify which organism is present, which organism is mapping to, best fitting to these reads. So these are all the results. Basically, with the laboratory technique, we found that Cryptococcus neoformans was found in all the 3 Caesar samples. But we in these three samples, we identified this many reads. 37,047 reads were generated in CSF1, and we found that 3,766 reads matching to Cryptococcus neoformans. So that proved that Cryptococcus neoformans is also present. We also found other organisms like Bartolleria and Homo sapiens also, obviously because it is from human. Homo sapiens also, some fields were mapping. Some more data also were generated. Some more species also were mapping, but based on like every sample will have any possible microorganism. So we'll get all of that, but what we are looking for is based on the disease type of this species is expected, and we look for it, and Cryptococcus neoformans is found with so many reads, and it is covering the genome completely. So similarly, the second sample also, Cryptococcus neoformans, was identified, and the third sample we found that Candida tropicalis is found, and it is matching with 22,799 genes supporting Candida tropicalis. So we proved that the conventional laboratory method is not accurate, and it can lead to errors. So, do I have time? Should I continue further?
Yes, ma'am, you can continue, ma'am.
Okay.
Yeah.
Okay, another project. This is the conclusion for this project that we have done nanopore sequencing and found that Cryptococcus was found in two samples, and Candida tropicalis is found in one sample. So based on that, the treatment has to be different, but actually based on laboratory technique, all the three were treated for Cryptococcus neoformans. So NGS is always advantageous. It gives accurate results. So another project, microbial diagnostics using whole genome sequencing approach. We have detected antimicrobial resistance. So antimicrobial resistance continues to be the major threat to public because in India especially, carbapenems are the important class of antibiotics that are reserved for severely sick people. The emergence of carbapenem resistance can result in outbreaks with limited treatment options, because we have very limited treatment options. The carbapenem resistance determining genes are VIM, IMP, and KPC, OXA, NDM. These are all if these genes, these are all plasmid mediated transfer into the species found, and then it becomes resistant to the drugs. So rapid detection of CIEs is critical, although the current methods are time consuming and are not efficient at determining the mechanism of resistance. So next generation sequencing methods helps, and I'm going to talk about that. So in this study, we have taken 65 reference isolates from NIH ATCC isolates from CDC panel, and then as a reference isolate, and 33 blinded clinical isolates from Johns Hopkins University, and 50 clinical isolates from Hinduja Hospital. And then we have done whole genome sequencing using MySeq machine with 100x coverage. That means every nucleotide is sequenced 100 times. So this is the methodology. So we have done lab-based tests also, and in WGS, whole genome sequencing, and also proteomics-based validation, particularly to identify the acquired resistant causing genes. We have done in the laboratory routine microbiology tests were used, like microscopic testing and drug resistance profiling were done. PCR-based resistance detection methods were used, and also whole genome sequencing and proteomics were used. So we do quality control of the raw data. We do the whole genome sequencing and quality control, and then reference-based alignment and de novo assembly. As I said, if the reference sequences are known, then we use a reference-based alignment, or else we do de novo assembly. So reference-based alignment against all possible bacterial species were carried out, and then de novo assembly also. There are tools that can use the sequences and assemble the sequences based on the overlapping sequences in the fragments. So there are tools to detect MLST, to do MLST, and what plasmids are present. and also what pathogen is present in this. Majorly, Kraken and Patrick are the two tools we have used for this analysis. And proteomics also we have done, and we isolate the sample and run it on LC-MS, and then PRM based analysis also done. So LC-MS is like it's a proteomic technique, mass spectrometry based proteomic technique. It identifies what are the proteins present in the sample. So we also identify which species it is. And PRM is a target analysis. Once we know what protein is present in which species, and we see the differential expression pattern using PRM. So basically to detect the acquired resistance. So these are all the results. We can see that JHU clinical isolates, there were 33, and so many were susceptible, and some of them are KPC positive. And the inside circular one shows the organisms present in it. It has Klebsiella pneumoniae, that's a major pathogen causes this drug resistance, and then Salmonella enterica and different species present in it. And similarly, clinical isolates from India, Hinduja Hospital also, we have found different what are the genes that drug resistance causing genes and pathogens present in it. And in the CDC panel also, we have done the similar analysis. And we also found several discordances. I can show you in table. So is it visible, my Word file?
No, ma'am, it is not visible, ma'am. Your PPT is only visible, ma'am.
I could not put this in the PPT, so I am showing it. Can you see now?
Yes, ma'am. We are able to see now.
So there are several discordances in the resistant markers. That means the gene, the resistance causing genes, and also the species identification. Comparing the clinical microbiology data with the whole genome sequencing data. So if you can see the first one, B1, B10, B19, these are the sample IDs, and then we have identified Klebsiella pneumoniae and KPC3 as the resistant marker, and in clinical microbiology data, and in whole genome sequencing data, we see that Klebsiella pneumoniae was found, but KPC2, another gene, was found, and proteomics results also shows that it is KPC2 but not KPC3. So it is a discordance of resistance marker with the clinical microbiology technique. And similarly, there are Enterobacter cloaceae and KPC were found, but in resistance in whole genome sequencing, we have found WIM1. So proteomics also showed WIM as the thing. So in the B19, WIM was found, but we found with the resistance marker with the whole genome sequencing, KPC2 was the resistance marker, and proteomics also shows that it is resistance marker. And for the B34, 37, and 39, NDM was found in clinical microbiology, and we found it is susceptible. There is no resistance marker found, and proteomics also did not show any resistance marker. So similarly, there are I54, 57, 58. This one, clinical microbiology shows susceptible, but we found NDM1. So these kind of discordances also can be efficiently detected using NGS technology. So discordant identified in a species. So in I06, E. coli was found, but in our analysis, Klebsiella pneumoniae was found. So like that, the species also can be discordant. And similarly, we have found both species and resistant markers discordance. Um, so in I13, Enterobacter species was found and susceptible, it is found to be susceptible. But we found Klebsiella pneumoniae and NDM1 OXA48 we found. So it's actually the pathogen is present and the patient may not be treated because the laboratory methods have detected that it is susceptible. In this project, we have shown different discordances comparing the clinical and NGS methods. So that's it. I'm winding up with this. So if you have any questions, you can ask.
Thank you, ma'am. Dear participants, now the session is open for discussion. You can ask your doubts to ma'am.
Good afternoon, baby ma'am. Very happy to hear your lecture again. This is
Yeah.
Sujata from Lady Doe College.
Yes, I
How are
can.
you, ma'am?
Yeah, I'm good. How are you?
Yes, ma'am. Fine. Ma'am, actually, it's very interesting to hear about microbial rich talk. I would like to ask you: is there any specific reason why you have gone for nanopore sequencing? Nanopore sequencing for like one of the microbial diagnostic research work, you have used nanopore. Other than low material requirement, is there any specific reason why you have gone for it, ma'am?
So first thing is we can instantly sequence immediately as soon as you isolate. You can just put it whatever DNA is present, it will generate. Second thing is it will generate larger fragments. That means it is more accurate. Means you can sure shot you can say that this species is present because in a single stretch it has generated the fragment. Nanopore sequencing, and also it is becoming very popular. It's a very small sequencer. You don't have to do. It's easy to sequence. Like on spot, you can do the sequencing and detect immediately. You can get the results because it is all like inbuilt programs are available, and it can generate the results instantly. So for the immediate detection of the pathogens, nanopore is mostly used.
Yeah, how will be the output form, ma'am? Is it like the same Sanger sequence electrophotogram, or it may be different from other the normal sequencing part?
It generates sequences. The output it gives is a basis, like sequences, how we get it from NCBI. Same thing we also get it. And then there are quality control and all, and then we convert it into our required formats.
While submitting the sequences like arose from these nanopore sequencing, should we mention the type of sequencing we have mentioned when while submitting our sequences to NCBI database, or not necessary, ma'am?
Yes, it is required because the you will get erroneous bases towards the end. So in the 3' end, you can trim it and then you can get the accurate sequence. But in case of the nanopore sequencing, it is more erroneous bases are generated. So that is the reason we have to mention, and whoever is using the data has to be very cautious on that.
So we need to specifically mention this sequences like we have got it through nanopore.
Yeah, yes.
Okay, baby ma'am. Thank you so much.
Yeah, thank you.
Good afternoon, ma'am. Hey, bless, ma'am. This is Mahalakshmi.
Hello.
Good afternoon. Hope you have health good, ma'am.
Yeah, I'm good. Yeah.
Okay.
Yeah, how are you?
Lecture was very good. I am also good, ma'am. I have a doubt. You had that there is a discordance in the identified species in
Yes.
the as well as resistant markers. In such a case, which is the most reliable one, ma'am?
Yeah, a next generation sequencing method is most reliable, and we
Okay.
suggest that both laboratory based techniques as well as next generation sequencing can be used for doubly check the diagnostics.
Okay. So
And
even then, if we do it in laboratory and then we do it in the sequencing, you are able to see a discrepancy. Then
yes,
which one the sequencing is most reliable, ma'am? That is what
yes, yeah, sequencing is most reliable.
reliable.
And
Okay.
also it is it's not laborious, and it's not time consuming, so
Mm.
immediately we can get the results quickly.
Okay, ma'am. Thank you, ma'am.
Yeah,
Your lecture was very useful, ma'am. Thank you.
yeah, thank you very much.
Wha-
Dr. Devi, sorry, Dr. Sripriya, you can proceed.
yeah,
Later I'll ask.
no problem, doctor. You can talk. I'll just ask after you.
Okay, just I'll ask only one more question, Dr. Baby Lakshmi. This is
Oh,
with reference to we are facing problem related to extreme drug resistance, multiple drug, MDR, XTR. These are very buzzwords nowadays. How are we going to overcome all these microbial resistance? Like even though we are using computational approaches, we are come up with so many different drugs, but still now we are facing this MDR and XDR. So what may be the possible ways we can overcome all those
so the major
unwanted things?
Yeah, the major drawback is it requires long time to culture these samples and detect that whether it is having MTB or not. So, because they have taken a long time, then the treatment is lapsed. So, because of that, we can the resistance keep changing, right? Resistance patterns keep changing. So they lead to XDR, MDR, and all. So if we can detect it immediately, the patient also come to the clinic as soon as they get the symptom, and also the given medicine they have to follow. They most of them do not take because of the side effects and all these issues. And early detected MTB are actually treatable, and it is it doesn't lead to the XDR.
Thank you, Dr. Baby Lakshmi. So prevention is better than cure.
Yes, yes.
Thank you.
Yeah.
Ma'am, I'm Dr. Sripriya. I just wanted to ask you something. So is it what is the kind of DNA we have to provide the company for doing the NGS if you're going to give it outside for sequencing? Will you have? Do you have any way, a very specific method of preparation of DNA, or you just give the microbes for doing the NGS samples just like that, or you have to make a DNA preparation and give them?
The sequencing service providers, they take even a sample, they do the DNA isolation and the prep the library and all. If you can do the DNA isolation by yourself, then they can take the DNA itself. So we have some kits, Guygen DNA isolation kits are available, and even there are the routine DNA isolation methods can be used, and we have to be very careful what we are looking at, and if we know the particular organism to be sequenced, then we have to isolate them and then culture them if needed, and then isolate the DNA and send it to the sequencing.
Ma'am, like how much of DNA, if you're going to give a DNA preparation, what is the amount of DNA? Is it less, or you have to give in a lot of DNA for
No,
sequencing, ma'am?
3 nanogram is sufficient
Oh,
for the sequencing. So we used to give around 1 ml, 1 mg of data, the DNA, and then they send it back, whatever. After they use it, they can send it back.
thank you so much, ma'am.
Yeah.
You're a very nice lecture. We could know a lot about next generation sequencing and its application yeah, in real time. Thank
Yeah.
you, ma'am.
Thank you very much, Shri.
Participants, you can come out with your doubts, please. Ma'am is there.
I hope they have not slept.
No, ma'am. No, they are all very attentive participants. If you people don't have any doubt, shall we wind up this session, ma'am?
Yes.
Yeah. Okay, thank you.
Thank you, ma'am, for your wonderful presentation.
Yeah.
A grateful heart is always a thankful heart. Now I request Dr. Shirley Cross, Assistant Professor of Zoology, to propose the vote of thanks.
Good afternoon to you all. On behalf of Marian Star Center, PG and Research Department of Zoology, St. Mary's College, Thoothukudi, Department of Zoology, Rani Anna Government College for Women, Tirunelveli, I am very delighted to propose a vote of thanks on this moment of occasion. I take this opportunity to express my sincere thanks to our chief guest, Dr. Devi Lakshmi Muthuswamy, Research Scientist, Institute of Bioinformatics, Bangalore, for her excellent presentation and making this session interesting and meaningful. Thank you, ma'am. As no program can become successful with a single person, I extend my big thanks to faculty participants for their support. Then I like to thank our Dr. Sister Principal, Dr. Sister Deputy Principal, Dr. Sister Overall Star College Coordinator, Controller of Examination, Deans of various schools, our beloved HODs, and all our faculty members. Thank you.
Thank you very much, ma'am.
Thank you, ma'am. It's a nice presentation. We appreciate your work, ma'am. It's
Thank
really a good service to humanity. Thank you, ma'am.
Yes, ma'am. Thank you very much. And thank you very much for all the participants. They are saying thank you. And in between, they have mentioned that audio is very low. I'm sorry, I didn't notice that. I hope it would make it.
Thank you, Dr. Baby Lakshmi. Wonderful lecture it was.
Yes, thank you. So, shall I leave the meeting?
Yeah, yes, ma'am.
Okay. Thank you very much for the wonderful opportunity, and I thank the organizing committee and Dr. Shri Priya for this for giving me this opportunity to present and have connect with you all. Thank you.
ma'am.
And stay safe. Bye.
Now we shall move on to the technical session on multiple sequence alignment and phylogenetic analysis. Now I hand over the session to Dr. A. Mahalakshmi. Mahalakshmi, ma'am?
Yes, ma'am. Thank you. Thank you. Am I audible? Am I
Yes,
audible?
ma'am, you are audible, ma'am.
Okay, ma'am. Can we give a brief quiz for the participants, ma'am? Before
Sure,
starting?
Dr.
Sure.
Mahalakshmi.
Sure, ma'am, sure.
Okay. I'll just paste the quiz link. I just wanted to know whether the participants are able to attend the quiz. 1 second. Yes, I have pasted the link. Participants, please let me know whether you are able to attend this link, this Google form. Yes, you are able to hear me, my dear teachers.
Yes, ma'am, you're audible.
Okay. I just wanted to know whether they are able to open the form.
Yes, it's open. It's open.
Thank you. Thank you. It's a very short quiz, just for me to know whether I'm able to communicate. It's not for you, it is for me.
Identify the database and then identify the protein sequence and plot for the database similarity.
Yes, ma'am. Your audio is Dr. Sri Priya ma'am, can we give them 10 minutes time? Because it just contains 10 questions. Sri Priya ma'am and Subhavati ma'am.
Yeah, sure, ma'am. We can provide
Yes.
them 10 minutes, ma'am. Yeah,
Okay.
sure, sure.
Yes,
Thank
Dr.
you.
Mahalakshmi.
Thank you. Ma'am, Sripriya ma'am and Shubhavadi ma'am, I got only 22 responses. What shall we do? Shall we start the session? Ma'am?
Yes, ma'am. Okay, ma'am. We can start.
We can start. Okay. We have got
Okay,
only
ma'am.
22 responses till now.
Okay, ma'am. Okay, ma'am.
So let them continue their quiz, ma'am. Shall I start the test quiz session?
Dr. Malak, please start the session.
Yes, ma'am. Yes, thank you. I hope we are in this weekend mood. But anyway, we'll finish the technical session in an enthusiastic and active participation as you have done in the last 2 days. Am I audible? Is my voice okay?
Yes, ma'am. You're audible, but the voice is breaking, ma'am.
Voice is breaking. Now it's okay? Now we are able to hear me clearly?
Yeah, it's okay, ma'am.
Yes.
It's okay.
Okay. Yes, just I'm planning on giving a short overview of what is that we have done and how this lab can be extended further. So it can includes multiple sequence alignment and phylogenetic analysis also. Yes, ma'am, I'll present a short PPT just for 15 to 20 minutes to give you an overview why the sequence analysis has to be done and what all things we can interpret using sequence analysis. Shall I present the screen, ma'am?
Yes, ma'am. Please proceed, ma'am.
Okay, thank you. Kindly let me know the screen.
Ma'am, your voice is breaking.
I'm able to hear you.
Yes, ma'am, but your voice is breaking.
Yes.
Yeah. Can you just check your network, please?
Okay, just I don't know why. It is high me. Now the voice is okay?
Now it's okay, ma'am.
Okay, thank you, ma'am. If you have any discrepancies, please let me know, ma'am. Maybe I'll be able to
Okay,
alter
ma'am.
it
Okay, we will
accordingly.
inform you. Okay, ma'am.
Okay, please, because if I am talking and you're not able to hear, it will not serve the purpose. Okay, thank you, ma'am. I'm just presenting a screen. Kindly let me know whether you are able to see the screen. Is you able to see the screen?
Yes, ma'am. It's visible, ma'am.
Okay. So here, the basic just a summary and an overview of what we have done. First day we have done a database search, and then we have went with the sequence database search. So here I just need to add the quotes of Theodosius Dobzhansky. Nothing in biology makes sense except in the light of evolution. Similarly, nothing in biology makes sense except in light of sequence comparison. So in today's lecture also you can see Baby Lakshmi ma'am was trying to compare the different sequences. So in bioinformatics, everything we start, we need to do a sequence comparison. Yesterday I mentioned 4 experiments. So second part of the experiment is comparison. So without comparison, we cannot do anything with bioinformatics. Okay. So this definition already we are aware about it. But anyway, for an overview, I'm just telling. Bioinformatics, the purpose of bioinformatics is not only collection, organization and analysis of large amounts of biological data using network of computers and databases. Normal terms, layman terms, people used to say bioinformatics is a marriage between a biology person and a computer science person, wherein they'll be able to collaborate their scientific knowledge and use it for the scientific community. Yes, so this is what I also yesterday showed to you. In today, we have seen how to find a retriever sequence, and then we are just going into the searching against using the computational analysis tools. As a few of the teachers, they showed their interest. Maybe in Tuesday's class, I'll be touching upon gene expression, and I'll also tell you how biomodels can be used. Okay, this is the workflow. So here I'm just trying to highlight whatever data that we use. When we use computational analysis tools only, we'll be able to get the meaningful information, and from the meaningful information, we'll be able to get the knowledge. So that is what we are trying to do in the lab. We are able to get the information and knowledge. Yes. So here this is an example. I have a sequence. You already have got a sequence. Okay. So here, this course, I just want you to note, there is a tremendous amount of information that is available in each sequence. And in the database, we know the database is enormously growing because there is a technological advancement in sequencing. Baby also mentioning all the latest techniques that can be used. So now we need, so the data is available as bioinformaticians or as biological or life scientists. We need to collect the data that has been available and try to collect the available data and try to collect the meaningful information. Yes. So this is what you have retrieved. This is called as the FASTA format of the sequence. What is this FASTA format? FASTA format is nothing but giving the symbol, greater than symbol, and then giving the sequence without any spaces. Okay, so here all of us are aware about it. So this 1-letter word alone, I wanted you to note, because when we do our analysis, we should be clear with an idea what is that 1-letter code. So that will help us to identify what is that amino acid and how it is helpful in the sequence structure function relationship. That is the core of bioinformatics. Am I audible, teachers?
You're audible, ma'am. Yes, you are audible.
We have a gene in our hand. The first question, whether I have any genes with a common ancestor. And for that question only, you have done a database similarity search. So when you do a database similarity search, I will be able to find out what are all the other genes that are close to my gene of interest. And then I also find out what is the function, similarity in function to other genes. Because I have an unknown sequence, I don't know what is that function. So what I do a database similarity search. So based on which sequence is similar, I interpret what is the function. That is the basis of doing our database similarity search. And then if I have a nucleotide fragment, I can translate and get the protein sequence, and we'll be proceeding with the protein sequence analysis also if time permits today. And I'm just trying to give you an overview. So imagine if you have done a new sequencing, in such a case, the tool you have to use a tool that is used for vector contamination, because I have cloned it in a gene and then I get the sequence. I shouldn't do the sequence analysis with the remaining of the vector sequence. So there is a tool which helps to check for, vector contamination. This is a tool that has been available. It is called as EMVec tool, wherein it's similar to your BLAST. I'll just paste the sequence that I'm having in my hand, but we'll check for vector contamination. As you have not taken it from the lab, we have not done that, but I'm trying to tell you because if we are doing it in a wet lab and we are generated it from our own study, in such a case, if I have got it from a vector, the sequence has to be removed because if the vector remaining sequence are available in the sequence, then that will give us all false results. That's the first step that you have to do. Yes, next slide. So here, this is a blast output. Here you can see the red lines indicate whatever sequence I have used, it contains vector contaminant. So this has to be removed. Only if I remove the vector sequence, I am sure the sequence that I'm having in my hand, my own query sequence. This I already explained. You are already aware about this database. You know, we are sitting in one place, then we are submitting our result to web server, NCBA blast, and then web server submits the query to the database server. So we don't know the interface that is happening. So here the database server returns to us, and then from the web server only we'll be getting the results. So this is a way how these databases are accessed, and these databases are accessed by different web servers, each offering a different interface. And this article yesterday I mentioned, all of us before we start to look into this issue. Look at the search data issue. It's published every year. Okay, so this is a tool that yesterday we mentioned. We have just used it to find similarities between sequences. So whenever I find similarities between sequences, we'll be able to find the function of newly sequenced genes. I'll be able to predict new members of the gene family and then explore evolutionary relationships. This type of you are very familiar. I'm not going to talk much about it. Okay, yes. This definition alone, I just wanted you to know. Yesterday, due to time lag, I couldn't explain this. We refer to the term called as score. Score is nothing but, as I already mentioned, it is the sum of the substitution. Substitution is based on match or mismatch in protein. Maybe in the when I explain the protein, I'll show you. Based on the substitution, they'll give a score. So if a substitution is of a similar property, it will be given a positive score. The substitution is of a different property. Say, for example, an acidic amino acid is replaced by a basic amino acid and next. So the score is nothing but the score is based on the alignment. What is the match and score? Okay, for proteins, they use two different matrices. I'm not going to talk much about it. Just for you to know. Yes. Is there any problem in it?
Dr. Maha, your voice is breaking and
there's some background sound also.
Yeah, there is also another background sound. Participants, please mute your mic. Doctor, I think it's Dr. V. Murugaya. You can proceed, Dr. Maha.
Yes, thank you. Am I audible now? Is my voice okay?
Yeah.
Yes, thank you. So yesterday we have seen about the score. The score is nothing but it is based on the matches and mismatches and gap score. For a protein alone, you have different types of matrix. I'm not going to talk much about it. I just give the explanation.
Mahalakshmi ma'am?
I think Dr. Mahalakshmi left.
She has left. Yeah, she has left, ma'am. Yeah, she has left.
Maybe a network problem. She'll come back. Please, participants, please wait for her. She'll come back.
Subha.
Ma'am.
Dr. Mahalakshmi.
Ashmi. Ma'am, she has left, ma'am. Now she has joined, ma'am.
Okay.
Screen visible, ma'am?
Yeah, yes, doctor.
Sorry, the network issue. No. So here you can see the E value, it refers what is the number of sequences that can occur by chance. As I already mentioned, when you have a low E value, it implies that sequence cannot occur by chance. It can occur only by, it is of high biological significance. Yes. So these two terms alone, I just wanted to mention. Indel. What is this indel? Indel is the insertion in one of the sequence and a deletion in the other sequence. I'll show it in the protein alignment also. I'll just show you. And then identity, I already mentioned. It is nothing but percentage of identical residues. Okay. So here, whenever we do a database search, we'll be able to identify homology. What homology? Say the two sequences are homologous. What do you mean by the term homologous? They can be homologous. That is, they can be similar, but if they have evolved from a common ancestor, I can say them as homologous. Say, for example, I have a part of a gene, and then the same gene is showing similarity to a bacteria. In such a case, I cannot say I am homologous to bacteria. But a part of the gene is showing similarity to apes, monkeys, gorillas. In such a case, I can say, as we have evolved from a common ancestor, I can say human sequences of homologs. Teachers, can I proceed?
Ma'am, your voice is breaking, ma'am.
Teachers?
Ma'am, your voice is breaking, ma'am.
1 second. Problem. Net is working only, ma'am.
I think your bandwidth is very low, ma'am. So that's what this issue is happening, I think.
Now it's okay. I have connected to some other net. Now it is okay. Able to hear my voice? Yes,
Now
tell
it's
this.
okay. Okay, ma'am.
Am
Now
I
it's
able to
okay.
hear? Okay. Okay. So now I just explained the E value and score. Is it okay? Can I move on to the next one? Next one.
Yes, ma'am, we can proceed.
Okay. Here, indel is insertion in one of the sequence and deletion in the other sequence. Today, basically, I'm also mentioning about indels. And then refer homology. We can say two sequences are homologous just based on similarity alone. We can say homologous provided they have evolved from a common ancestor. That's what I'm saying. We can say I am homologous to apes, monkeys, and gorillas, not homologous to bacteria. Because they are not even from a common ancestor. Can I move to the next slide now? Am I clear? Yes.
Ma'am, your
So this is the
slides
way
are not.
how a sequence is being represented. Not shared. Thank you, ma'am. Thank you. Yes, now you are able to see my slide.
Yes, ma'am. Yes.
Now you are seeing my slide. Yes. Sorry for the inconvenience caused. I explained the E value, Z score, everything is clear. Can I move on? That is more important. Ma'am?
Yes, yes, ma'am.
Yes. So here, this part alone, I just wanted to highlight. So these numbers refer to what is that region that is aligned. So here we understand from the query 421 to 654 is aligning, and it is entirely aligning. It is showing 100% identity. Okay. So here you can see a gap from 654 to 753. It is not aligning. That means that region, both the sequences are different. So that is referred to as an indel. There will be some gap given so that from 753 onwards, the same sequence is aligning. I hope you are able to get me up, able to follow me. So here you have a query sequence. The query is aligning from 421 to 654. And then here from 753 alone, the query sequence is aligning. So here you can understand there is a gap, there is a region in between. That region is not aligning. So all this, we can get the details from the alignment that we are doing. Is it clear, teachers? Yes. Can I proceed? Okay. So here, yesterday also I showed you, here also I'm trying to highlight. Here you see there is a gap, and then here I'm showing a base pair. So this is called as frame shift or SNP. SNP is your single nucleotide polymorphism. So here you can see this is also called as indel, insertion in one of the sequence and a deletion in the other sequence. So here I'm just highlighted a region that I showed you yesterday also. So based on this only, say for example, if you're working on SNPs, I'll be just looking at whether there is some base pair change that I can locate. So this same blast can be used for different purposes depending upon our own interest, our own goal. We can't say it's just a blast, just doing a database. We have to look into the details. Looking into the inner details helps us to do the purpose that we want. Yes. So here you can see here it has been someone asked me yesterday how to do a genomic blast. So this is an example wherein I have just taken the nucleotide sequence and I have done a genomic blast. So here genomic blast is I have a gene, I just do a blast against all the genomes that are available. So here when I do like that, it is able to show that my gene is aligning with the musculus chromosome. So I can do a genomic blast to see where in the genome my gene is located. Okay. Yes. So here, this is all of you are very familiar. Flat file, I'm not going to talk much about it. Okay. Now we move on to the next aspect of our multiple sequence alignment. So here, multiple sequence alignment, the main purpose of doing this multiple sequence alignment is these 4 main goals. In order to detect conserved motifs, do a phylogenetic study, and then prediction of functional residues and secondary structure. This is the purpose of doing multiple sequence alignment. As we already know, multiple sequence alignment is when we try to align more than 2 sequences, is called multiple sequence alignment. My students, whenever I used to handle my classes for my students, if I say just 2, they'll come, I'll take 3 and finish lab. I'll advise them, no, take just 2 sequences. The more the number of sequences you take, the more you'll be able to understand what is the conservation pattern. Okay, can I move on a little more, teachers? Yes. So this tool have you here and I just showed what are all the action numbers. This is
ma'am, your voice is again breaking. Ma'am, your voice is breaking.
1 second. Sorry. Sorry for the inconvenience. Now it's okay. Now it is audible. Teachers, now it is okay. Able to hear me?
Yes, Maha.
Yes, ma'am.
It's going on fine. In
Yes,
between,
yes,
actually, your voice is breaking, Maha.
I don't know. I don't know why there's a problem. Internet connection unstable. But
otherwise,
anyway, I'll
it's
check it with the Geo.
Yes. But I have one question,
thank
Maha.
you.
Before,
So this is
before
yes, ma'am.
proceed further, I would like to ask one thing. You said like a number of sequences to be taken for multiple sequence alignment. Will you specify how many number of sequences should each one will take for this MSA?
change the different okay features.
Now it's okay.
Okay, sorry. I changed a different location. It's not network. Maybe that area net is not coming, I believe. Now it is okay. Able to hear me? Okay.
Yes, ma'am.
So now
Yes, ma'am. Yes,
proceeding
ma'am.
with the- Sorry for the inconvenience. I couldn't help you. So here, this is the tool that we are going for the lab, Clustal Omega. I'll demonstrate in the lab also how to do. I'm just giving an overview. So this is how your results will be. So here, can you see the slide that I projected, teachers?
Yes, ma'am.
Yes.
Sujata ma'am had some doubt. Sujata ma'am, she was asking and I just
Yeah,
had an internet issue.
Yeah,
Sujata
I have
ma'am,
asked
please.
about. Yeah, I have asked about like you were mentioned about number of sequences to be pasted or to be used for MSA. So will you please mention how many number of
Yes,
sequences would any like whoever is wish to do MSA, number of sequences to be taken from the better database?
Normally, as per our own professor, Professor Krishnasamy, he used to mention, to ask, try to based on the content. We have more sequences 100%. We cannot be all those. We can do sequences.
Ma'am, please,
So
voice
that
is
we
completely
able to
breaking, ma'am.
Voice breaking.
Maha, do
Like-
one thing, Maha, do one thing. You can just turn
switch
off
off your
your
video.
laptop. No. Better you can turn off your video as well as instead of using laptop. Why don't you try with your mobile, Maha? It would be better. Maybe you can send the PPT to Marian Center. They will show you the slide sharing. You can use your mobile. Will you please try in that way? I think it will work.
Okay. It will work.
So you can pass the PPT to Dr. Shripriya or Subhavati ma'am. They will share your screen.
Yes, one second. So before I do that, can I ask the blast protein alone demonstrate and do? So let them continue. Meanwhile, I'll send it to Shripriya ma'am.
Yeah, now the voice is clear now.
Now it's clear. அப்படியே continue பண்ணு.
இதே position.
Yes. Don't change the position. Yes.
So,
video off pannir. Maha, you can turn off your video, I think.
1 second. Sorry for the inconvenience.
No problem. It's all the problem is very simple.
Yeah, this is all
thank you.
expected. Yes.
Okay. So now, as she asked, I cannot take just 100% sequences. The more the number we take, the more we'll be able to get meaningful information. So what I advise my students normally is I'll ask them to take different ranges of sequences, 100%, 99%, 98%. அந்த மாதிரி எடுக்க சொன்னோம்னா we'll be able to identify what is the conservation pattern seen among those organisms. I answered your question, ma'am.
Okay, Maha. Proceed. இப்படியே proceed பண்ணிரு. Okay.
So now you are able to see the multiple sequence alignment file. The slide is visible for you, teachers?
Yes.
Is it visible? Yes.
Yes,
So here you can see some double dot star. It's okay. Able to view it. At the bottom, they are given symbols: a star and a double dot. What does the star indicate? Star is conserved. It is seen throughout the same. Maybe when you do a multiple sequence alignment, I just wanted you to observe this. Star indicates.
Please go for slide sharing. It's
okay. Now it's
full
okay.
screen.
Now it's okay.
Yeah, it's fine. Full screen.
Double dot refers to it is there is a substitution, but the substitution is of a similar property. Yesterday I was mentioning, no. So normally in evolution, if it is of an important region, nature what it does, it permits variation, but the variation is of a similar group. Say for example, an acidic amino acid is there. In such a case, an acidic amino acid will be replaced. That is what we mean by conserved substitution. So here, when you look for double dot, the double dot indicates there is a change, but the change is of a similar property. But if I see star, the star refers to conserved. Conserved is irrespective of whatever evolution line it is conserved throughout. So that is very important because that gives us a clue that is very essential for the sequence structure function relationship. Am I clear, teachers? Yes.
Yes.
Can I move on next point? Yes. So here there is some coloring given. The coloring is being given based on the property. So here blue refers to acidic amino acids. So here you can see blue is being the acidic amino acids, and then magenta color refers to basic amino acids. So here you can see wherever I kept double dots, the double dots, there is a change, but the change is of a similar amino group, amino acid. It's of a basic amino acid that is being replaced. And then coming to the green refers to hydroxyl group, and then the others are given as gray. So here, just in multiple sequence alignment, what is your lab that you're going to do is you're just going to do the multiple sequence alignment. I'll demonstrate, and then you'll be just observing for the star and the double dot. Maybe you can paste that image in the Word, and then you can highlight what is the conserved region and what is the semi-conserved region. Yes, that's a tool called ClusterLex. You're not going to use it. I'm just trying to tell you that can also be used. It's a downloadable software. And then this is the way how a sequence conservation should be represented. Teachers, are able to see the sequence logo? Any guess you can say by looking at this PPT? Teachers? Sequence logo?
What do you mean by sequence logo?
Sequence logo is based on the sequence conservation.
Yes, ma'am.
We represent
now only we
the
see.
sequence. So
Yes,
here,
yes.
Yeah, now only we can see.
see. Yes, you can see the height of the column is proportional to how it is conserved. If I say full G, that means in that particular position, there's no other base pair. But in certain places, I'm giving 4 letters. That means, you see, for example, in 17th position, capital T is higher, then comes K, then comes P. So here, sequence logo is that's some tool that can be used to represent the way the sequence is represented. This is another way of representing the consensus. Conserved sequence. So normally, when you have a conserved sequence, we can write just like that. But sometimes you will not get one base pair or one amino acid. In such a case, you can represent like this way. So the greater height indicates it is predominant or it is seen at a higher frequency. Can I go to the next slide? So the bottom one refers to the position and the y-axis refers to the frequency. Am I clear?
Yes.
Teachers? Yes. In the
Ma'am, excuse me. When you say the colors, the red color represents what? Blue, magenta, green, and
that color is not here. They're given a different coloring. This coloring can be varied. Only for the multiple sequence alignment, I mentioned that this doesn't have that meaning. Here it's just a coloring that is being given. Only in the multiple sequence alignment, they use those colors.
Okay, thank you.
So coming to the for our lab, you'll be using today. You'll be doing multiple sequence alignment, and then you'll also be doing the phylogenetic tree. So here I've just shown it for my own sequence. So here this is my LDC9 strain. It is showing clustering with this particular isolate and showing this value. What is this value is? This is the bootstrapping value. What is this bootstrapping value? Similar to the E value, this bootstrapping gives the reliability or reliability of the tree. So when I'm able to see 100 times, what does it mean is what they do in this bootstrapping is they slice all the multiple sequence alignments. For an example, I'm saying I have just 10 base pair for a multiple sequence alignment. So what I do, I take 10 sequences and I do a multiple sequence alignment. After they get the result, for each multiple sequence alignment, they'll construct a phylogenetic tree. So here it is trying to jumble up. Enna பண்ணுவாங்க? They try to reshuffle first, second, third, fourth, fifth, sixth, seventh position. So each base pair will be reshuffled, and then for each reshuffling, they'll do a phylogenetic tree. So this number tells me they have done 100 times reshuffling, and for every 100 times, they were able to see this clustering is same. Am I clear? Am I clear with this bootstrapping?
Yes.
Yes. So here you understand they are trying to jumble up. They are trying to reshuffle the alignment, and then for every reshuffling, they'll construct a phylogenetic tree, and they will count how many times this clustering is seen. So when I'm able to see a higher bootstrapping value, that indicates that clustering is highly reliable. Okay. Yes. So now with this, I'll move on to the lab session, because next proteins, I thought I can finish the protein sequence, but first we'll finish the lab part. I'll stop presenting this. 1 second. You share. I'll go to the protein that I'm having in my hand. 1 second. Teachers, you are able to see this result? Blast results?
Yes, Maha.
Yes, ma'am. Yes, thank you. Thank you. So here, as I gave you the homework for Blast P, were all of you able to do the Blast P, teachers? Any problems you have? Teachers? Please respond.
No, ma'am.
So the same way as we do our Blast N, thank you. At the same way as we do our Blast N, here what we do, we just click on Blast P, just paste the protein sequence. So here, for the explanation purpose, I have done and kept this ready. So I've got the results. So here, it is showing different percentage of identity with the query sequence. Okay. So here, I just wanted to show the alignment also. There will be a minor difference in the alignment. So here, yesterday I mentioned there will be a vertical line in the sequence alignment. Here, can someone tell me what is the difference between this protein sequence alignment and nucleotide sequence alignment? Pictures? You're able to see the alignment that I projected?
No vertical line in between the sequences.
Yes, yes. There is no vertical line, but here in between, they are just given the letters. So here you can see when they give the letters, that indicates it is identical. So in some places, you will not be able to see the lines. In some places, you will be able to see plus symbol. What does that plus symbol indicate? Teachers? What does that plus symbol indicate? You are able to see a plus. It's not a match.
Addition
Here,
of.
here you should understand, it is not a match. Here you see there is a substitution. So here, in the presence of isoleucine, valine is seen. So plus refers to substitution, a similar substitution. Yes, when there is no plus symbol, it indicates it's a mismatch. Am I clear? Yes. Teachers able to follow me? Serine and alanine, because they belong to the same group of amino acids. But F and S comes, it is phenylalanine and serine. Here they don't give a plus symbol. That plus symbol, if it's absent, it indicates it's a mismatch. I mean, the plus refers to similar. This is what I'm trying to say, similarity and identity. Teachers able to follow me?
Yes, yes.
Yes.
Ma'am,
So,
please once again repeat, ma'am, please.
One second. So here you have all letters in between. Okay. When you have all letters in between, the letters are there.
Okay,
in some places, you are able to see it. Yes, ma'am. Able to hear me?
Again breaking, madam.
Your voice is breaking.
Again, let's see. It's okay. Now the voice is okay.
Yeah, fine.
Yes, yes.
Sorry. So here you can see there is a plus symbol in between. I'm highlighting a particular region. You're able to see it, teachers?
No.
You're able to see the highlight.
No, you are not able to see.
No, well, I have a share screen. No. One. Sorry.
You are sharing, but we are not able to see your highlight.
Now we are able to see.
No, Maha, your slide is okay, Maha, but your voice is breaking in between. Like you need to repeat the content, like whatever you mentioned about the plus symbol. You just make sure that your network is okay, and then please repeat the content again.
Okay. Okay. Now am I hearing?
No, Maha, not yet.
It's okay.
No, still is not audible clearly.
Now it is clear. Teachers?
Yes, Maha. Yes.
Okay. Now I'm sharing that screen. You able to see my screen? Blast result. Yes. Yes. So here I highlighted a plus symbol. Able to see it?
Yes, ma'am. Yes, yes.
Yes. So here I'll repeat. Please bear with me. I don't know. I couldn't help this internet. I don't know. So here in between, you are able to see the letters. The letters indicate they are identical. Am I clear with this, teachers?
Yes, ma'am. Yes,
Yes.
ma'am.
But here you can see same line. I can see. I highlighted the entire line. Here you can see in between, you are able to see plus symbols. Teachers able to see this? I highlighted
Yes,
a
ma'am. Yes, ma'am.
particular line. So that plus symbol indicate in the presence of Q, there is K. In the presence of V, there is L. So there is a plus symbol indicated. You might ask me, why is that plus symbol indicate? The plus symbol indicate they belong to similar matches. So as I already mentioned, the score is based on whether they belong to similar amino acids or different amino acids. So here, when they belong to the same amino acid group, a similar amino acid group, they'll be given a different score. When they belong to very different amino acid contrasting property, the score will differ. That matrix itself is a very big lesson. I'm not touching much about it. I'm just giving a clue about it. What is that I'm saying? If the amino acid is being replaced and the replacement is of a similar amino acid. For example, valine is being replaced by leucine. They belong to the same group. Okay, non-aliphatic R group. In such a case, what they do, they give a score plus score. Say, for example, in the presence of valine, a leucine comes. Originally, if it is 10, what they give it as 8. Okay, but in the presence of valine, something else comes. What they give a negative score. For example, aliphatic aromatic amino acid on this negative score. Am I clear? Able to follow this?
Yes, ma'am.
Yes, ma'am.
Similarly, in certain places, you are not given a plus symbol. Can someone tell me what do you mean by this? I'm highlighting a particular region. You're able to see? In certain regions, you are not able to, no plus symbol is given.
That's a deletion.
No, it's not a deletion, my dear. The deletion, it is a mismatch. Deletion, they'll give a hyphen. So here you can see there is P and L. When I say deletion, in one of the sequence, they put a dash. That is what it's called deletion. Here, that is a base pair, but it is a mismatch. Able to understand? P is proline and L is your leucine. Okay. So as they belong to different group of amino acid, no plus symbol is given. Am I clear? Teachers?
Oh, yes, ma'am.
Yes, ma'am. Yes.
So I'll give one more thing. 1 second. I'll just change the way it's represented. So here you're able to see the alignment view I'm showing, teachers. In the blast, you have an option called as alignment view. I'm just changing pairwise with dots for identity. Now you'll be able to appreciate it clearly. Now you're able to see, teachers?
Yes, ma'am.
Yes. So here, wherever dots come, it indicates it is identical. Okay. So, but here at the bottom, you can see, you're able to see? You're able to see some red letters?
Yes.
The
Yes,
red letter
yes, ma'am.
indicates it is different. Okay. So here, in this, you can see identity. Identity is the same. Positive refers to similarity. Gaps I already mentioned. Is it clear? So only in the protein sequence alignment, we'll come across this concept of positive. I hope you understand what is positive. They give positive based on whether they belong to the same amino acid. If it belongs to aliphatic group, they'll give a plus. If it belongs to positively charged amino acid, both are positively charged amino acid, they give a plus. Both are negatively charged amino acid, they give a plus. Both are polar, uncharged groups, they'll give a plus. Am I clear?
Yes.
Yes. So the scoring is based on, so here they represent. So whenever I talk about protein, I shouldn't give identity alone. I should also talk about similarity. This positive is nothing but similarity. So here you can see it is 98% identical and 98% similar. Is it clear? Teachers?
Yes, ma'am.
Yes. So you can understand when it is identical, it will have a higher score. See, for example, when it is having 100%, I'll conclude this, then you'll have to move to blast. So you can see here 100%, it is 1177 because when it is totally the same base pair, the score will be different. But you can come down on just one thing I'll show. For here, you can see this is 1154. So here you should understand when the amino acids differ, there is a different scoring pattern. So அதுக்கு என்ன score ma'am கொடுக்கணும், அது எப்படி நமக்கு தெரியும் அப்படின்னா, we are not going to give the score. There is an algorithm which will do that. But as scientists, as teachers, we should be in a position to understand how that scoring is being given. The scoring is being given based on amino acids of similar property. So when there is an amino acid of a similar property, for example, so simple example சொல்றேன், ஒரு valine இருக்கு வந்து 10 score கொடுக்குதுன்னு வெச்சுக்கோங்க. அந்த valine இருக்க இடத்துல isoleucine வருது அப்படினா அதுக்கு 8 score இருக்கும். ஆனா valine இருக்க இடத்துல ஒரு aspartic acidோ இல்ல வேற ஏதோ aromatic amino acidோ வந்துச்சுனா அங்க score வந்து நெகட்டிவா மாறிடும். Got it? Able to understand what I am saying? Teachers?
Yes, ma'am.
Okay. So now we move on to the multiple sequence alignment. Yes, ma'am. Voice okay? Teachers?
Yes, ma'am. Okay, ma'am.
Okay. So now, what we are going to do is here at the top, you are able to see the web page that I am projecting. In the blast page, in the end, you are able to see the download option that I am highlighting. The mouse.
You could see.
Yes. Able to see? Yes. So what I am doing is, I don't want the complete sequence. Complete sequence na, some of the bases will be very large. What I'm trying to take is, I'm going to take just aligned sequences alone. So what you are going to do, just click on download FASTA aligned sequences. சரியா, நான் ஏற்கனவே இந்த மாதிரி டவுன்லோட் பண்ணி வச்சிட்டேன். ஓகே, நீங்க பார்க்கணும் அதுக்காக I'm just doing it. Sorry. So here it will be downloaded in the folder. Teachers, you are able to see the notepad that I am presenting?
No.
No? One second. Let me present that. Okay. Now you are able to see the notepad?
No.
Now I am sharing
Yes,
it?
yes. Now it's.
Yes, sir? Okay. So whatever I have downloaded, this is available. Okay. Now what I'm doing is I'm going to cluster omega. So here, just for your understanding, here you shouldn't give large names like this. So first 3 sequences alone I'm going to do. For your homework, you're going to do lot more number of sequences. Just for an explanation I'm doing, don't give the same name. Say for example, I give Bacillus thuringiensis 9, Bacillus thuringiensis 10. The software will say more than 2 sequences with the same name. So what you should give is, you try to give some short names so that it will recognize. So I'm giving BT1. So நம்ம என்ன பண்ணனும்னா, இன்னொரு டேபிள்ல parallel BT1 na enna accession number அப்படின்றத நம்ம நோட் பண்ணிட்டே வரணும். So that later if you have any clarifications, we can go to that reference number and see. சரியா? So here I'm just giving BT35. I hope you are able to understand what I'm trying to say. You are able to see the correction that I'm making? Teachers, the notepad I'm doing something.
Yes, ma'am.
Yes. So here you don't give lengthy name. Don't give Mahalakshmi 1. It will say all sequences the same name because it recognizes only 8 characters. So pinnadi na enna number potalum software recognize pannadu. I'm just giving 35 because that was a number that has been given. சரியா, ஒரு 3 மட்டும் தான் பண்றேன். Just for time, conserving time. For yourself, you have to take more number of sequences. Try to take sequences with different percentage of identity. இப்ப என்னோடதுல வந்து 100%, 98, 97 தான் இருந்துச்சு. உங்களோடதுல எல்லாமே 100% இருந்துச்சு அப்படின்னா, you cannot see the semi conserved. So try to take sequences with differing percentage of identity. Yes. So it has large, all the sequences has to be taken. For time purpose, I'm just giving a few. Is it okay? Teachers?
Yes.
Yes. So I'm just copying sequences alone. I have to do the same alteration for all. I'm giving you as a homework. I'm just copying it. I'm just going to the new share. I'm going to Google. Yes. You're able to see the Google page that I'm doing, teachers?
Yes, ma'am.
Yes. So just type cluster omega. Cluster omega, what you're going to do is, you're just going to, I have just copied 3 sequences. I'm just pasting it here. So if you are doing it for a nucleotide, you have to change it as nucleotide. If you're doing it for a protein, you can change it as protein. Is it okay? Teachers,
Yes.
able to follow me? Yes. So here, this is the symbol that will recognize. So whenever you paste, all the sequences should have this, and it should have a different name. Say for example, I type BT11. I say BT all. It will say all sequences the same name. So you should give different names so that it will recognize it. Is it okay? Teachers, able to follow my concept?
Yes, ma'am. Yes.
Yes. Okay, I just pasted the sequences. And then it gives cluster W with character counts. Everything keep it as default. Just submit your job. Or you can notify by mail also. I'm just submitting a job. Teachers are able to see the process. Your job is currently running, Varda.
Yes,
You're
ma'am.
able to see the web page? Yes. Okay. So here you'll be getting, yes, you got very quick, you're a bit lucky. We have got the results. So here you can see, as I just taken only 3 sequences, yes, I'm able to see some double dots also. Yes, double dots indicates it is of a similar semi-conserved substitution. Okay. When there is no dots, it indicates it is a variant residue. Okay, here you are able to see some gaps, teachers.
Yes.
Yes, there is a gap. And then you are able to see some single dots. Single dots refers to weak similarity. Double dots vandu konjam higher similarity. Single dots vandu konjam weak similarity. When you are not able to see any symbol, it indicates it's highly variable. Am I clear? Maybe you can just take a view of this, put it in a notepad, and then you can highlight indicating what is the conserved, what is the variable. It is for you to remember later. Yes, teachers? Can I move on to the next step? So with this, we conclude the multiple sequence alignment. Now I'm taking you to the phylogenetic tree. So download alignment file. Yes, I just downloaded the alignment file. So what I'm going to do is I'm going to take you to the tree top. I'm just copying this file. Entire file I should copy. I'm just copying the file. I'm taking you to the new share. Share. One second. Go to the Google. One second, I'll open and then I'll come. Copied it. Let me copy the alignment file. Just copying the file. I'm just copying and pasting it in the notepad for my own reference. Problem I need not do it again. I'm just copying it. Now what I'm doing is I'm going to Google. Teachers, you're able to see the screen that I'm putting?
Yes, ma'am.
Yes, you're able to see the tree top?
Yeah.
Yes, thank you. So here I just clicked on Gene B. This is a very easy, user friendly. That's why I've taken this particular software. Yes. So here we have tree top. Teachers are able to see the link that I'm taking. Tree top phylogenetic tree prediction. So here what you are going to do, you are just going to copy the alignment file. Don't copy the input file. Just copying the alignment file. I hope you know what is an alignment file. That is after we get our input. Okay. So here you have to remove this letter. Cluster omega, multiple sequence alignment. It has to be removed. Teachers are able to see my demo?
Yes, ma'am.
Yes, thank you. So here we have to give the bootstrapping value. Only if we give the bootstrapping value, you will get the number. I'm just submitting it. It will take some time. Okay, so it is working. Yes, we're very lucky, Panama. We have got the results immediately. Yes. Yes, you're able to see, teachers. So here it says BT1
Yes, ma'am.
and
Yes.
BT35 is clustered together. As I given 3 sequences, it is just giving for 3. So whatever we feed in, it will be given as output. So you can't say multiple sequence alignment. If I just use 3, then my 3 will not be a valuable information. If I use only 3 for my multiple sequence alignment also, it will not be valuable. So in order to increase the content of information, we normally have to go for larger number of sequences and larger number of sequences of differing percentage of identity. Just don't say I have taken 20 sequences with all 100% identity. Then all will be conserved. Is it clear, teachers? Yes? Teachers?
Yes, Dr. Ma. Yeah,
One more point alone. I mean, this I'll wind up. Monday lab will be going for protein structure prediction. So I'll just show you a website and then we'll end the session for protein sequence analysis. 1 second. Stop sharing. Till now, any doubts you have, teachers? Any doubts you have?
No, ma'am. It's clear.
It's clear. Okay. I'm just presenting another screen just for you to explore later, because Monday we'll not be talking much about sequence analysis, protein sequence analysis. We have to link both structure prediction and what to say? Yes, Monday we have to finish structure prediction and docking. So here today I thought I can just show you an overview of protein sequence analysis. Maybe Monday I'll start with what are all the analysis are very essential, because I don't want to give a detailed lecture now. I'll just show you that website alone just for you to note. Teachers are able to see the sequence analysis page. This you can note down. This gives a detailed information of protein sequence analysis. Teachers are able to see this web page.
Yes, in silico analysis.
So here you can see it is based on our own interest. So it gives for also, it gives for immunological interest also. So here we can use it for, I'm just moving down. I'm not taking much of your time. We can use it for protein identification. If I have a sequence, I can use it for protein identification. I can go for primary structure analysis, compute PI or molecular weight. And then I can also go for multiple sequence alignment. You already are very familiar with that. And then I also go for pattern and profile searches. So all this, I'll just give a brief overview on Monday before I go with structure prediction and docking. What are all the sequence analysis tool and how it can be used. As it's the last hour of this session, I don't want to touch much about it. Maybe I will share this link. Maybe I'll copy this link and paste it for you. Maybe you can later explore depending upon your own interest. It's a very useful site, which I found it very interesting. It has all aspects, all in one-man shop. Just 1 second. Teachers, I pasted the link. Kindly note down this website. You can use it for your protein sequence analysis. Yes, any other doubts, teachers? Sorry for the inconvenience. I don't know. I couldn't help you. There was a network issue again. I have 2 net
That's
and all
okay.
are 5
That's
GB.
okay, Dr. Maha. Don't feel like internet problem is always there.
Any other doubts you have related to this lab session?
Ma'am, excuse me. Is there any limitation to submit the sequences number?
Ma'am, I'm not able to get, ma'am. Please, ma'am. Please repeat your question, ma'am.
Is any limitations for the number of sequences to submit?
No, ma'am.
No.
No, ma'am, there is no limitation. We can go with individual submission. Say, for example, I have 1 or 2 sequences. I can go with individual submission. And there is another tool which permits for bulk submissions. I can go for bulk submissions also. There is no limitations of how many sequences a person has to be submitted. The idea of receiving it is they wanted it for the scientific community. The only problem they have is we have to submit a scientifically validated annotated data. That is what GenBank or EMBL or whatever databases we are submitting, they'll be just looking for whether the data that we submit is valid.
Okay. Thank you,
okay. Have I answered your question, ma'am?
Yes, ma'am. Yes, thank you.
Yes. If time permits, I'll also show you how sequences can be submitted. I'll just give a short view. The workshop is very tight. So we are trying to accommodate the participants' interest. Maybe I'll give you a brief overview of what are all the tools that can be used, but we cannot proceed with submission.
But if you were explaining that, that will be very useful for us, ma'am.
Yes, ma'am. Yes, ma'am.
We are doing project work at that.
Okay, ma'am. Maybe we can try a short demo.
Okay, ma'am. Okay. Thank you very much.
Thank you. Thank you, ma'am. Any other doubts, ma'am? Teachers? I thank all the teachers for the positive encouragement. I had a network issue, but even then you are saying it's wonderful. Thank you, useful. Thank you so much. Any other specific doubts, teachers? Okay. If you have no doubts, I hand over the session to the organizing team, Dr. Sri Priya ma'am and other Subhavadi ma'am. Yes, maybe Monday class, I will be giving a brief Google form test for the multiple sequence alignment, phylogeny, and some protein sequence analysis. Is it okay, participants?
Yes.
Yes. Thank you. Sorry for testing the teachers. I have no right to test the teachers. It's just for me to know whether I am able to communicate.
Thank you, Dr. Mahalakshmi. It was a wonderful session. Wonderful session. And the way you have been trying to make things clear was really nice. And all the things, whatever the small things you were explaining to us was really nice. And I think we could very simply do all this multiple sequence alignment and phylogenetic tree construction. I think this is all very basic and very important for teachers to know, so that they can handle such classes, practical classes for students, UG and PG students of their colleges. Thank you, Dr. Mahalakshmi, for this session. And please don't worry about the internet connectivity. It is a universal problem. Everybody, I think, everybody will know about it, and there is no issue, Dr. Mahalakshmi. Thank you for sharing your expertise with us. And we are waiting for the next session, and we are eagerly waiting for it. Thank you, Dr. Mahalakshmi.
Okay, ma'am. With the organizing team permission, one of the participants has asked for GenBank submission. When can we have it, ma'am? On Monday? Monday, it will be very tight. Sequence analysis, structure, prediction, docking, and then we need to include a GenBank submission also.
How long will it be enough that when you have 1.5 hours, Dr. Mahalakshmi, on that day? Because we can ask Dr. Ilakyam to cut short her talk to half an hour.
Okay, you can talk to Ilakyam, ma'am. Then we'll decide.
Yeah, I'll talk to Dr. Ilakyam.
Inform me accordingly. Otherwise, I'll give a GenBank submission stepwise. I'll put it in a PPT and just show them.
But I think GenBank submission will be really nice for people to know, Dr. Mahalakshmi. It will be the basic thing that. Right. I think, shall we have it on? I think Monday will be the right time. Monday we'll have it, Dr. Mahalakshmi. Even if you have to skip one of the molecular docking or something like that, we can take it to the next day.
But the participants had some other option, no? They wanted that
and that will take the whole day.
That has to be explained a bit. One of the participants expressed interest in the array express and then the biomodel.
Then we'll talk to Dr. Ilakyam about it, Dr. Mahalakshmi.
Okay. Thank you. Thank you for the opportunity. I thank the organizing team for giving me this opportunity. I thank all the teachers. Even though it is a weekend, Saturday, all our teachers are so active and they are so listening and asking questions. I feel happy. Thank you.
On behalf of the Department of Zoology, St. Mary's College, and Department of Zoology, Rani College, I would like to thank all the participants and the team of faculties who have been arranging this. Thank you all for your patient listening and for being with us. Thank you all. Dr. Shubhavati, Dr. Mahari, you can leave the meeting. Shall we close the meeting, Dr. Shubha?
Yes, ma'am. We shall wind up the meeting. Yes,
yes. Thank you.
Thank you, Dr. Mahari.
Thank you.